Mutations. Sometimes things don't go as they're supposed to in cell replication. Gene mutations and mutations in the number of chromosomes can occur. So this is to do with 3.4.3. Genetic diversity can arise as a result of mutation or during meiosis. So this is from the AQA specification. Gene mutations involve a change in the base sequence of chromosomes. They can arise spontaneously during DNA replication and include base deletion and base substitution. Due to the degenerate nature of the genetic code, not all base substitutions cause a change in the sequence of encoded amino acids. Mutagenic agents can increase the rate of gene mutation. Mutations in the number of chromosomes can arise spontaneously by chromosome non-disjunction during meiosis. Gene mutations involve a change in the DNA base sequence of chromosomes. The types of errors that can occur include substitution. This is where one base is substituted with another. For example, ATG-CCT becomes ATT-CCT. So the G is swapped for a T. Another example you can see here, it goes GCT-CAG-AGG in the original gene, but in the mutated gene, the C is swapped for an A. So it's substituted for an A. So then when that codes for an amino acid, the GAT codes for a different amino acid, it codes for aspartic acid. So the original protein would be alanine, glutamine, arginine, but the mutated protein would be aspartic acid, glutamine and arginine. Or you can have deletion mutations. So one base is deleted. For example, ATG-CCT becomes AT-CCT. The G has been removed. These tend to be more severe deleted mutations. So you can see here there's an original gene, TAT, AGT, CTT. But then here there's the base A is deleted. So that means there's a frame shift. So every triplet after that might be different. So it goes TAT, then GTC, and then that's it because there's no triplet there. So instead of the original protein being tyrosine, serine, leucine, it'll just be tyrosine and then valine. The order of DNA bases in a gene determines the order of amino acid in a particular protein. If a mutation occurs in the gene, the sequence of amino acids it codes for and the protein formed could be altered. The degenerate nature of the genetic code means some amino acids are coded for by more than one DNA triplet. Like tyrosine can be coded for by TAT or TAC in DNA. Therefore, not all substitution mutations will result in a change to the amino acid sequence. Some substitutions will still code for the same amino acid. So here, the T is substituted for a C, but TAT and TAC still both code for tyrosine. So although there's a mutation, it still produces the same protein. Substitution mutations won't always lead to change in the amino acid sequence, but deletions will always make a change. The deletion of a base will change the number of bases present, which will cause a frame shift in all the base triplets after it. Mutagenic agents. Mutations occur spontaneously, for example, when DNA is misread during replication. However, some things can increase the rate of mutations. These are called mutagenic agents. And examples are UV radiation, ionizing radiation, some chemicals, and some viruses. Mutagenic agents increase the probability of a mutation occurring. For example, there's always a chance of gene mutations occurring. However, under normal circumstances, the probability of a mutation happening is very low. This probability is increased by exposure to mutagenic agents, such as UV radiation. The likelihood of a mutation occurring somewhere is increased when an organism is exposed to UV radiation. Chromosome mutations. In humans, when meiosis works properly, all 4 daughter cells will end up with 23 chromosomes, one from each homologous pair. But sometimes meiosis goes wrong, and the cells produced contain variations in the numbers of whole chromosomes or parts of chromosomes. For example, 2 cells might have 23 whole chromosomes, one each of 1 to 23, but the other 2 might get a bit muddled up, one having 2 chromosome 6s and the other having no chromosome 6s. This is called chromosome mutation and is caused by errors during meiosis. Chromosome mutations lead to inherited conditions because the errors are present in the gametes. One type of chromosome mutation is called chromosome non-disjunction. It's a failure of the chromosomes to separate properly. So Down syndrome is caused by a person having an extra copy of chromosome 21, or sometimes just an extra copy of part of the chromosome 21. Non-disjunction means that chromosome 21 fails to separate properly during meiosis. So one cell ends up getting an extra copy of 21 and another gets none. When the gamete with the extra copy fuses with another gamete after fertilisation, the resulting zygote will have 3 copies of chromosome 21. Explain what a mutation is and how mutations can have no effect or a positive effect. Changing the base. A mutation is a change in the base sequence. This results in the formation of a new allele. Because the genetic code is degenerate, so amino acid sequences may not change at all, or a mutation in an intron, so the amino acid sequence may not change, or a description of how it is degenerate, so some amino acids have more than one triplet code. So it could change the amino acid sequence but has no effect on the tertiary structure. Or the new allele is recessive, so it doesn't influence the phenotype. If it has a positive effect, it could result in a change in the polypeptide that positively changes the properties of the protein. It may result in increased reproductive success and may result in increased survival. A summary: gene mutations involve a change in the base sequence of chromosomes. They can arise spontaneously during DNA replication and include base deletion and base substitution. Due to the degenerate nature of the genetic code, not all base substitutions cause a change in the sequence of encoded amino acids. Mutagenic agents can increase the rate of gene mutation. Mutations in the number of chromosomes can arise spontaneously by chromosome non-disjunction during meiosis.